husbandry have always been the backbone of our economy. However, we often overlook the untapped potential of pig farming as a serious business venture. Today, with scientific methods, improved reach, and growing market demand for pork and pork products, pig farming is no longer a marginal activity, it is a profitable enterprise. I am sure that the talk will be profitable and fruitful for everyone present here. I welcome you all once again to the event and hope that you all will enjoy the talk. Thank you. Thank you.
Thank you so much, sir, for your welcome speech. Thank you, sir. Now I request our coordinator, ma'am, Dr. Sangeeta Das, to say a few words. Thank you, ma'am.
Thank you, Zakaria. Respected Principal, Bahana College, Dr. Sandeep Saiha sir. Respected our today's resource person, Dr. Rajiv Dey sir. Thank you so much, sir, for accepting our invitation and being with us on this platform today. My research fellows, Monika and Zakaria, and all the dignitaries and participants across this country. Institutional Dietech Hub, it has been continued in our institute since 2012, and in 2023, under phase 2 project of Department of Biotechnology, we initiated this online lecture series on entrepreneurship. Today is the 8th session or episode of this lecture series, for which we have with us Dr. Rajiv Dey sir with us. And I believe this talk will be fruitful for all of us, especially for the young person. And I will consider this lecture program as a fruitful one if we can receive some feedback, maybe in terms of some academic collaboration or maybe in research. Then only we will consider that this lecture program has been fruitful for all of us. So with these few words, I welcome our resource person, today's resource person, and all the participants to this online lecture program. And with this, I conclude here. Over to Zakaria.
Thank you so much, ma'am. Now I request Ms. Malika Barua, Laboratory Assistant Biotech Hub, to introduce our resource person against everyone.
Thank you, Zakaria. It's my privilege to introduce Dr. Rajiv Dey, who is currently serving as senior scientist in immunobiotechnology in the Indian Council of Agriculture. Presently posted at ICAR National Research Institute. BDSC, PhD degree, bringing 13 years of experience in diagnosis and vaccine development. He has completed a prestigious postdoctoral fellowship under the ICAR Postdoctoral Fellowship, India, TWS Fellowship Program, and ICMR DHR International Fellowship UK. Dr. Dey's prolific academic contributions include 57 research publications in peer-reviewed journals, 6 books, and 6 patents granted with 5 patents claims alongside the development of 6 technologies. His work has been widely recognized through numerous awards and fellowships, including the International Fellowship for Young Biomedical Scientists, INSA Associate Fellowship, NAAS Associate, NASA Associate, INYAS Membership, NABF Young Scientist Award, and the SCRB Young Scientist Grant Award. He has been actively involved in projects funded by DBT, ESA, SCRB, EAFD, and ICAR. We are delighted to have Dr. Dey with us today and look forward to his valuable insights.
Thank you so much, Ms. Malika Barua. Now I request our today's chief guest to start the meeting. Thank you, sir.
Maximum volume.
Sir, am I audible?
Oh,
Yes, sir.
just a minute, sorry, network issue. Okay,
Okay, sir.
so first of all, I would like to thanks to the coordinators and all the faculty members for nominating me for today's interactions with all of you. I'm sorry that some, I think, internet issue, so that I don't know my voice is audible to all of you or not properly. So anyway, it is a nice interaction. So let us start my presentation. So I'll be sharing my slide. Is this slide is visible?
Yes, sir.
Okay, fine. And you see it is going forward or not?
Yes, sir.
Okay, fine. So, okay. So we'll be starting here. So the presentation which has been assigned to me, it is on the recent advancement in diagnosis and development of vaccine candidates against swine viral diseases, that is the piggery industries. This is a part of our animal science. So I also thanks to the coordinator for introducing me. So no need to introduce about myself again. So I'm basically now currently working as a senior scientist at Indian Council of Agriculture Research, and I have been posted at National Research Center on Pig, and which is located on the Rani. You know that it is near the airport and that Assam. And you can sometimes you can visit also. We have a wonderful laboratories and all these facilities is there. So before starting my presentations, I think all of you know about our Indian Council of Agriculture Research, right? So Indian Council of Agriculture Research is an autonomous council which is under coming to the Ministry of Agriculture and Farmer Welfare. So our ICR, it has been divided into different sections. If you see, it has coming from crop science, horticulture science, natural resource management division. We have agriculture engineering. We have fishery, agriculture education, agriculture extension, and one of the important section is the animal science. So where I have been posted and I have joined as agriculture research scientist in animal science. Right? In so far, we have 19 animal science institutes across the India. So this is the 19 Animal Science Institute. It has been located in all over the parts of our country. Okay, so some parts like it is depend on the where those animals or livestock population is higher. So for example, if you see the UP, then your Haryana and all these regions, we have institute for cattle, Central Institute for Research on Cattle. I have been posted there before. Then we have National Dairy Research Institute. We have the Institute for National Research Center on Buffalo, goat. Okay, so like this, it is depend on the part. Like Rajasthan part, there is also Camel Research Institute is there. Then Sheep Research Institute is there. Okay, so some other institute we have the Indian Veterinary Research Institute. It is the only two deemed university in animal science. One is our Indian Veterinary Research Institute and our National Dairy Research Institute, and both from there the many students getting degree from ICR in animal science, and they have been distributed in different parts of the country, different institutes. So our institute, it is located in the northeast. If you see, we have that population of pigs, we have population of yak, we have population of raccoon. So based on that, the three animal research institute it is located. One is in the Arunachal Pradesh, that is ICR National Research Center on yak. It is in the Dhirang. It is sorry. So ICR National Research Center on Mithun. It is located on the Nagaland. And our ICR National Research Center on pig, it is located in our Guwahati, Assam, where I have been posted currently.
So in this way, this all the animal science institute has been distributed from the in India. So there are total 19 animal science institute. There is some institute for poultry also, some others for buffalo, camels, and all these things. Okay. So now coming to my institute, that is ICR National Research Center on Pig. It is located at the Rani. It is in the near to the Meghalaya border. You know that. So this is a beautiful campus we have beautiful laboratories. We have all sorts of facility, starting from your normal laboratories to biosafety laboratories. Then we are regularly giving trainings to students, farmers, other stakeholders in different aspects. We have wonderful farm facility. We have wonderful this board semen collection center. Okay, so many. Sometimes you can visit and you can meet all the scientists here. So, and there are different disciplines of scientists that are available there, starting from biotechnology, biochemistry, physiology, and all kinds of facility. And now we are also working on the cutting edge research in the swines, in the piggeries, right? So, let us see about the livestock. I think some of you have known about that. So far in India, that livestock is contribute 30.47% of total gross domestic values in the agriculture sector, right? Followed by its contribute 4.1% GDP, and overall GDP is 4.1%, and livestock contribute among total agriculture. If you see, livestock contribute 25.6% of total agriculture GDP. However, overall Indian GDP, livestock contribute 4.11%. In agriculture, it contributes 25.6%. So, employment, you see that employment to about 8.8% of the population, you know that. So far, 8.8%. Excuse me. If you see, 20.5 million people so far in India, their livelihood totally depend on the livestock, right? And if you see that among the livestock regarding the piggery sector, okay, so it is also play a very important role. Among the livestock, the highest livestock which share contribute in the GDP, it is the cattle, which contribute at least more than 35%. For if you can see, buffalo, it contributes more than 20%, sheep contribute 30.83%, goat contribute 27.74%, and the piggery sector, it contributes 1.69% to the total livestock GDP. Because you know that the piggery, they are rare only for the meat purpose. And there also pig meat, there is certain religious that
আমি খবর নিয়ে পাই দিই কানেক্ট কৰিব মনে কৈছে নি এইটো দি সেই লেপটপটো কানেক্ট হৈ গ'ল
right? So far, pig production is in the current year, it is the pig population in India. It is only 9 million. It is 9 million. Okay. This may be due to that the pig products they are exporting somewhere. Some may be due to some management or some other issue. Okay. So if you see, out of 9 million pig population in India, 45% of pig population present our seven sister constituted northeastern region, right? So 45% it is located in the northeastern region. So out of 9 million, around 3.94 million, or you can say around 4 million pig population is present in north. It is the as per the report of DAS in 2009. So now coming to briefly about the piggery sector in India. So you know that the pigs are increasingly rare by marginal tribal communities, which signifies their roles in the rural Africa as well as the livelihood, right? So I've already mentioned that pig population, they are mostly concentrated in the eastern and northeastern region. In the eastern region, it is in 2.8 million, and in the northeastern region, it is 3.95 million states. Among that, highest population in our Assam, which is 2 million states, followed by Uttar Pradesh, it is 1.35 million, West Bengal, it is 0.82 million. Jharkhand, it is 0.73 million. And Nagaland, it is 0.7 million. Right? So, 70% of our Indian pig population, which is reared under your traditional smallholder, low input demand driven production system. However, there is also certain semi-commercial pig farms also available nowadays. It has been arising in Kerala, Punjab, and Goa states is this. Okay. Now this typical production system of these pigs, mostly what they are using, it is very easy to rear the pigs because simple pigsty you can use, some kitchen waste they can give for their feeding. Okay, some integrated piggery also it is possible with the what to say fish pond and all. So because that pig guns and all it can be consumed by fishes and from there means it could be an integrated system that possible. Okay, so however, what are the major constraint or difficulty in the piggery in India? So these are the most important. One, you know that social taboo. Many people don't like to consume the pork. Even it has been posted, I am also not eating the pork. Okay, so this is because I am a scientist, I have to do some research. That is another issue. Okay, so social taboo is there. So another is the availability of the germplasm. Getting the semen or those germplasm also sometimes it is very difficult. So it is distributing also it is difficult. And most important thing is the diseases. Because animals are always fond of getting various kinds of diseases, like some of the diseases of the piggery, like PCV, that is porcine circovirus, CSF, classical swine fever virus, porcine parvovirus, Japanese encephalitis. You know that recently many people have died due to JAB in Assam. I read in the newspaper because that JAB also spreading from mosquito, from pig to human. So, and there is another disease, PRRS, that is porcine reproductive syndrome. And the most important disease in piggery nowadays it is called ASF, that is African swine fever, which has been arised in the year 2020 from China. Okay, so these are defined diseases which went out. So if you classify the diseases can be classified in various ways. There is, like you can say, there is emerging diseases and transboundary diseases. Now, what is the difference between emerging and transboundary diseases? Right? Emerging diseases mean these are new diseases which was not existing previously. Okay, which is occurring in an area or host population which are spread further and get established in want of awareness. And this is why, because there are no diagnostic facilities available against that, because the disease is newly arrived, right? And the expertise or other factor is not there. That is the reason they are called as emergency. But disease of past, if they are re-occurring in an area having susceptible host population, they are called as re-emerging diseases. So emerging means totally new diseases, and re-emerging means if some disease was there very early, okay, and after that there is lots of care, it was not present, but immediately suddenly it has been sparked up. So these are called your re-emerging. And another disease is called your transboundary diseases. So this is the most important and common diseases in case of piggery sector, transboundary diseases. These are the animal diseases, okay, which are epidemic diseases. There are two terms. One is epidemic, one is endemic. Okay, endemic means all over. Epidemic means certainly it is there. Okay, that are highly contagious, okay, or transmissible and have the potential for rapid spread across geographical boundaries. Because of that, there is geographical boundaries. Okay, which may come to the different state or not state, different country to the India. So these are called transboundary diseases, which leads to you severe economic losses as well as it may also assist for the public health. So what the transboundary animal diseases can cause? It can cause the direct economic loss, which will reduce the animal production, reproductive failure, adversely affect the food safety, human health. Okay, it can threaten the food and nutrition security. Okay, that may also cost effectiveness for livestock management. So these are the drawbacks of your diseases, emerging and transboundary diseases. Now coming to what are the possible sources of transboundary diseases in pigs especially. So there are different sources. For example, migration of animals. You have migrated the animals from one country to another. Migration, you know that there are certain migratory birds that can carry certain viruses, bacteria, and can spread from one species to another. Okay, then maybe also natural spread by certain insect actor. Okay, or through wind also it can be transmitted. Then impact of your contaminated biological or okay, or like human being itself infected peoples, contaminated animal byproducts which are you are transmitting, illegal trafficking of the animals. You have seen that some of the from India transporting some of the animal products or agricultural products in some other countries like USA, UK, it is very difficult because they will scrutinize all the products whether it is when it is transmitted. Sometimes they are not accepting also. So in this thing there is certain legal issues. So illegal trafficking it is another important, like from China, if it is transmitting some of the pork products, meat products or something. So this is also a transport transmissible transmission, right? Some others like climate change, microbial distribution, food waste from international aircrafts or ships. These are the reasons why the transboundary disease is being transmitted. Right? Now, why the India are prone to getting the transboundary animal disease? You know that India, it having the land of around more than 50,000 kilometers, okay, and then having a coastline area, it is around 7,560 kilometers. India shared border of the 9 countries. You know that different 9 countries it is border like Bhutan, Nepal, China, okay, Myanmar, and all these things. Okay, Bangladesh, okay, with a 7 and sea border. So this is the reason why there is always chance to coming the diseases from neighboring countries to the India. Okay, northeastern region, they raises around 47% of the country, and it has border. You know that northeast has border with Bangladesh, it has border with China, it has border China, East region, Arunachal Pradesh, Bhutan, okay, and Myanmar is at the boundary of the. So, in pigs, which are rare in India, they are more susceptible to various emerging diseases because it may easily transmitted from China and all these reasons. Like for example, I have already mentioned the most important disease in India which is arising is African swine fever or ASF, and another is CSF, classical swine fever, GRRS, FMD, swine hepatitis, and smell. Due to its porous international border in the neighboring country. Okay. So what are the defined transboundary diseases of swine in India? So African swine fever, classical swine fever, horse and reproductive and respiratory syndrome virus, PCV2 that is called post-weaning multisystemic wasting syndrome, foot and mouth disease, and some other club hepatitis. So briefly I'll talk about the each diseases. So it is not so relevant to you because you are molecular diagnostic. But first you know that some diseases which are important for piggery sector. So one is important disease is foot and reproductive and respiratory syndrome. This was first identified in the year 1991 in Netherlands. Okay, it is an RNA virus. Okay, it is a positive stance RNA virus, and it is caused by your ITVVD family. It having 2 genotypes. One is genotype 1, that is European genotype, and genotype 2 is North American genotype. And they are these 2 genotypes of TRRS. They are antigenically they are diagnosed. Understood? Means these 2 genotypes they are failed. So it is in that genotype 2 of these TRRS, they are more dangerous than the genotype 1 because they cause the severe respiratory diseases. Okay, the virus it can be present in the fecal material, urine sample, semen sample of the pig. Okay, it can spread through the fomites formation, which include the vehicle, insect bites, and all this. Okay, so the disease has been identified 1987 in United States and America. Later, it has came to the Asia, Europe, and North American countries. So another disease is your SMAD, that is your stillbirth mummified embryo death infertility, which is caused by porcine parvovirus. This is also an important disease. Okay, against this PCV, PRRS, what I told, there is no vaccine available in India so far. Similarly to this PPV, that is porcine parvovirus, Okay, this is against that also. There is no virus, although the vaccine is available in outside, like modified live vaccines and all. But India so far no vaccines is available at this time. So in India, PPV alone or combination with other potent circovirus was first identified in 2010. What it is causes? It causes embryonic death, infertility in pigs. Okay, so these are the most positive factors for this PPD or SMARTY. Another disease, it is important disease, and I am working also on this angle is PCV2, that is your porcine circovirus. You know that circovirus is the smallest virus, right? And it is a DNA virus. So this is called multi-wasting disease because it causes severe losses because PCV is the one of the important virus which causes immunosuppression. You know that if the body they are immunosuppressed, not immunocompromised. So what will happen? There will be chances for getting other diseases also. Okay, other bacteria and viruses easily can infect that animal and causes severe diseases. So PCV2 it is the most common disease and it is uniquely present. If you screen the pigs everywhere, the PCV2 will be present. So this is the most important. So you need to manage. And this is also so far the vaccine we are taking from outside, from abroad to a sarcoplex, but now it is discontinued. So our team, we are working on this vaccine. So I will be talking in the later slide. So for porcine circovirus. Okay, so this is the, in endemic area what happened, routine immunization against mostly what they were doing before, they are giving the vaccine. In India only one tick vaccine available, that is in classical swine fever virus. That is sometimes they are using to control the disease. But sometimes it is seen that CSF vaccine is not effective against the PC, leads to outbreak of the disease. Okay. So another important disease, you know that it is called foot and mouth disease. Because why? The disease is the infection will be present in the foot or mouth disease. This is called FMD. So FMD it is caused by Picorna virus, which is the most important contagious disease. It can be spread by contact between the animals. It having multi-serotype, that is 4 serotype, serotype O, A, C and ACO1. Okay, it is present in India, and India is one of the countries with the largest population livestock. With that 528 animals, they are found to be susceptible so far report of FMD. Among that, 11 million pig we are included. So FMD vaccine which are using now is Raxa FMD vaccine. That is the specific for cattle, that one only using for the pig. Provax, Raxa, they are using in India for prevention of the FMD. Okay, so under this it is classical swine fever, right? So classical swine fever, this is also a highly contagious transboundary disease of domestic pig and quail. Okay, this is also a transboundary disease. This is belong to Pestis C within the family of Lepidopteran. The transmission of this virus generally occurs between the pig through direct contact or mechanical by mites. Okay, so first it was reported in Telugu in 1833 in USA, followed by in India it was first reported in 1944. Okay, in district in Uttar Pradesh. It was the first reported in India in Alibag. After that, the disease began to spread other areas of the country. Most frequently reported now it is northeastern region. It is more common. Okay, so this is what are the causes of this classical swine fever. The animal, there will be high fever, there will be constipation, diarrhea. These are the common symptoms. Okay, and the animal will die also. In India, that is our Indian Veterinary Research Institute, which is located in Jaffnagar, Uttar Pradesh. They developed a vaccine called Raxa Plus against the classical swine fever vaccine. It's the live attenuated vaccine and which is available in India for prevention of classical swine fever. So the most important disease it is in pig it is nowadays African swine fever because it causes 100% mortality of the animal. So you can imagine that there is huge losses to the farmers. Okay, but we are very much unlucky so far. Why? Because we don't have vaccine against the African swine fever. Now also recently reported in the Arunachal Pradesh, our team has visited and collected samples. Means there is lots of spreading to the African swine fever in India, especially in the northeast region. So this African swine fever virus possessive ASF, it is a large envelope double-stranded DNA virus, okay, and it is under the family called Aspergillus. So African swine fever, it can be transmitted either direct or indirect contact. It is not only direct, indirectly also it can come. Okay, please, or it may transmitted that sometimes it has reported that some soft ticks with some actor that is under the family of ornithopods. They are also responsible for disease the ticks. Okay, and responsible for spreading the virus. Okay, so this African swine fever, it was first reported in 1921 in Canada. Okay, in 2020, I told you, May 2020, India first epidemic report of African was in Arunachal Pradesh. It was reported. Okay, and why it was investigated through China, that is in Siam River, Brahmaputra River, some of the dead animal carcasses entered India, and from there they spread it. Okay, so later it was reported that the diseases spread in several other states, including Assam, Mizoram, Andhra Pradesh, Gujarat, Himachal Pradesh, and West Bengal. It causes hemorrhagic fever with high mortality rate. You can see more than it is 100%. If one animal is infected in a farm, there is all the animals will means 100% animal is infected. This is the early mortality rate. There are some common early clinical presentations like high fever, inappetite, yes, and the animal will get certain death. Okay, so this is the most clinical sign. So pig can also present with diarrhea, vomiting, dark, and all these things. But why we are unlucky in India, not only India, worldwide also, there is no vaccine, approved vaccine is available. Because why? Because there is so many reasons. Because you know that African swine fever virus, it is a very complicated genome is there. Okay, so scientists are unable to find so far which are the genes responsible for giving the protection. Okay, so it needs lots of work. So this is the reason the vaccine not yet added. And there are also 24 genotypes available. So some genotypes they are again mutating also. You have seen for COVID also, right? So similarly for ASF, similar strategy is going. So the vaccines, although we are also working on this angle. So we are also collaborating with some UK, some other institute, and let's see that things are arrested. Okay, so compared to other diseases, the diseases both detrimentally affect social, economic, and India has been reported that so far Indian 2.76 billion has been economic loss due to swine. So, as we don't have vaccine, what are the measures for this prevention and control of the ASF vaccine? So, the most important that you should go for biosecurity measures. So, this is the most important. Strict biosecurity, we need to go to the farms, we need to avoid the farmers, we need to avoid with some of the local distributors who are associated with the big salaries or for production. Okay, practice slaughtering, and the most important that biosecurity we have to, and we should go for diagnosis of this animal having ASF. So early diagnosis is most important for any diseases who don't have vaccines so far. Because if you diagnose first, so you can take some biosecurity measures that will follow this strategy to prevent the spreading of the disease. So there need to be a disease surveillance and monitoring system. So what are the challenges against this ASF? So I already told you this is a transboundary disease. It can be transboundary spread. It can process the water, the movement of liabilities or cost or contamination. It causes a severe economic loss. It affects the consumption. Okay, it affects the impact of the livelihood of the farmer. Okay, so early detection of ASF is critical, but surveillance and diagnosis it is needs to be done properly for preventing the diseases. And so far we don't have any common vaccines against ASF. So biosecurity is the only method. And another challenges is that lack of awareness. Because some pig farmers think that if the animals is infected to ASF, it will come to them also. If that you assume that what the virus will enter, but no, because the ASF virus is not known. Okay. consuming this infected pig also it cannot transmit, but it can spread to his own farm. So he should avoid those consuming those infected animals properly. Okay, and some other is that some wild bulls which are present in the jungles and all, they are using as an example of that case. Although they will not show any symptoms, but they will be simple as an example. They can spread to the domestic animals. Okay, this is another. So there should be phase-trade restrictions should be there among the countries for transmitting some pork products or something. Okay. Then socio-economical disparities should be there, inconsistent regulated framework should be there. So these are the challenges it is facing in the case. So why there is constantly the vaccine development in ANZ? Because there is unknown protective immune response, because the full immunity which the virus it is implanting, the host is not yet known. Okay, and there is also the immune evasion strategy these viruses follow. So this is another constant. Okay, protective immunity against the viral protein. I know that this ASF genomic is a very complex. So which are the protein responsible for giving to that protection is not yet known. Excuse me. So this leads to difficulty in production of your subunit vaccine. Okay. Now this successfully developed for low virulence strain is commonly available. So these are the cross protection studies also very limited. So these are the constraints why the vaccine also are not developed in the African swine fever virus. Okay. So now coming to that is the reason we should go for proper diagnosis. That is called as disease, animal disease surveillance system in India. So what happened? The Government of India having bifurcated into Ministry of Fisheries, Animal Husbandry and Dairy and Ministry of Agriculture and Farmer Welfare. Okay. Under this Ministry of Fisheries, Animal Husbandry and Dairy, there is a department called DASD, that is the Department of Animal Husbandry and Dairy. Okay. And they have under that there is State Animal Husbandry Department, State Animal Husbandry Department is there. Then from there again Animal Husbandry Department and district level also there. And among each district there is veterinary dispensary hospital is there. Okay, under that there is certain animal health program or regional disease diagnosis laboratories is available. And under the Ministry of Agriculture and Farmer Welfare, we have the Department of Agriculture Research Education here. Under that our Indian Council of Agriculture Research coming. And I have already mentioned under ICF we have that Animal Science Division. And it has been distributed in different states. We have that Animal Health Institute defined laboratory which are working on the system. So this is all about how this animal disease surveillance system in India. Okay. So diseases are working on defined important diseases like FMD, brucellosis, classical swine fever, pestitis, pathogens, ruminantis. Okay. So far in India, if you see, 12,000 veterinary hospitals is available, 25,000 veterinary dispensary available, 28,000 veterinary aid center available, which are regularly working on animal disease surveillance system. Okay. So there is also lots of disease diagnostic laboratories at different state and district levels are available, which is certain disease diagnostic laboratories. So this is about that how this India, what are the animal disease surveillance system is working. So now coming to our main topic, that is the diagnosis and vaccine. So let us see about the diagnosis. So all of you know what is diagnosis, right? If you go to the diagnosis, what is the first you should know? What is the difference between a diagnostic test and a screening test? So, means screening test is what? It will identify asymptomatic individual who may have the disease. In the airline individual, they don't have the disease, but they are not asymptomatic. So, if you go for certain screening, so this is called screening. You know that commonly screening tests, like you might have known that for cervical dysplasia or cervical cancer, if it is the doctor is suspecting that, he will say go for pap smear test. For heart diseases, we have to go for fasting blood cholesterol, and you know that for diabetes, what we have to do? We have to take the fasting blood sugar. Okay, hypertension, we have to measure our blood pressure. Breast cancer, we are going for mammography. For prostate cancer, prostate we are going for PSA test. Okay, brucellosis we are going for MRT. Glaucoma, eye we are going for ocular pressure. If you have hyperthyroidism, we are going for detecting TSS. So these are all common screening methods. Okay, these are you are not going for a symptomatic patient, but in an asymptomatic individual, we are regularly full body checkup. So some tests we are doing. So this is called screening. And what is diagnostic test? Diagnostic test it is used to determine the presence or absence of a disease when a subject shows sign or symptom of the disease. Once the individual it is showing the symptom, then we wanted to know it is due to which disease. So for that we are going for your what is called diagnostic. So the diagnostic test it is performed after positive screening test only. Okay, first we have to go for screening test, then we have to go for diagnostic test to establish a definitive diagnosis. Okay, so now diagnostic test it has been categorized by the World Organization for Animal Health (OIE). Okay, that is the main central agency worldwide which are reflecting that which diagnostic test should be used. So it has been described diagnostic test as a prescribed test and alternative. What is the prescribed test? These are those tests which are considered optimal for determining the health status of animal before shipment or reporting the diseases. Means these are the tests which has been well adapted or well tested by the all over the world, and they have prescribed that these are the tests is written. These are called prescribed. And alternative tests means what? They do not demonstrate the absence of the infection in the tested animal with the same level of confidence as prescribed. Means alternative tests tell so yes it may be infected or may not, but prescribed tests tell definitely these are infected. However, the OIE Terrestrial Animal Health Standard Commission considered that alternative tests chosen by mutual agreement. There should be an agreement between the importing and exporting countries. Okay. For giving valuable information for evaluating the risk of any of the animal or animal microorganism. Okay, so now coming to what are the rules for selecting which diagnostic test you will follow. So there is a thumb rule, that is rule out and rule instead. Okay. The selection of an appropriate diagnostic test is depends upon the intended use of the result. Means what purpose you are going to use that result. Understood? So if your intention is that rule out of the disease, what is the rule out? Means you wanted to then whatever the negative result is there, you want to identify that these are the negative results. So if you mention that this is the rule out of the disease, which is reliable negative results are required for which the test having highly sensitive. That is, sensitivity indicate there are few false negatives, means all are positive. So you have to take it out that which are not negative, which are not positive. Okay, so this is called rule out. And what is rule in? If you desire to confirm a diagnosis or find evidence of a disease, that is rule in the disease, we require the test which are reliable positive result. That is high sensitivity. So high sensitivity means it is few false negative, and your rule in means highly specific. That means your reliable positive result. So this is the two intention for diagnosis. One is rule out and one is rule in. Okay. So what is the thumb rule? It is given by Pfeiffer. So a test, if you consider a test having a 95% sensitivity and 75% specificity, so it should be used for the rule out of a disease, right? And if you consider 95% that opposite, specificity and 75% sensitivity, so it is rule in for a disease. So this is the thumb rule for selecting a diagnostic test. It is rule out and rule in based on your specificity and sensitivity. And specificity and sensitivity, they are the heart of any developing a diagnosis. So specificity and sensitivity, for example, suppose you have infected for COVID Okay, so you got fever, you got some headache and all these things. Okay, it may be due to common cold virus infection. Okay, so specifically if you want to know it is due to COVID, not for common cold, you have to determine this is the means specificity. Means because this is because of only the COVID, not for the other common cold or related virus. So this is specificity. And sensitivity means what how much it is sensitive. Okay, it may give sometimes false negative result. Okay, means at the lowest condition up till where the virus can be infected and cause the disease. So these are specificity and sensitivity. They are the heart of the disease. Now, for evaluation of a diagnostic test, okay, it requires some independent elite method tests which are known as gold standard. Gold standard means EHT, well-established diagnostic, okay, which are regularly used for diagnosis of a disease. Now, this gold standard, they may be a single unequivocal test. For example, it may be single test, histological test, postmortem test. Okay, suppose postmortem lesion you got, or a combination of different alternative tests also. Okay, means it's not only a single, it may be a combination of different. But sometimes what happens, if certain gold standard test doesn't exist, a particular condition, there should be some diagnostic should be developed and it should have level of agreement between different tests. Okay, so then only we can agree with yes, test can be utilized. So this assumes that agreement between the test is evidence of the validity with the how much the test is valid. Where this is disagreement means tests are not valid, is not reliable. So we should not use those tags. Okay. So the characteristics of the test of the gold standard should be considered based on its specificity and sensitivity. So now I already mentioned specificity and sensitivity is the heart and brain of a diagnostic development. Now how can you define the sensitivity and specificity? Sensitivity can be defined. It is the ability of the test, okay, to identify directly those who have the diseases. So here you consider A is the disease. From all individuals, the diseases. Okay, mean A is the perfectly they have the disease, and all individuals which are susceptible for the disease, they are A plus B. So sensitivity is A divided by A plus B, means true positive divided by total disease spread. Okay, this is your sensitivity. On the other hand, specificity, it is the ability of the test to identify correctly those who don't have the diseases considered it is from all individuals which are free from the diseases like B plus. So the specificity B by B plus C means true negative divided by disease free means there is no disease. So this is the way how we can calculate. So this is an example it is given. So we must know the correct disease status correct calculation is that gold standard test is the best test available. It is often, I already mentioned, it may be invasive or non-invasive. Okay, and the new test which you develop, it is a new screening test or maybe which should be rapid, should be a less expensive test. Okay, it should not be so costly also. So how you will compare? Suppose you got this is the it is a new test we developed in laboratory and it is the gold standard. So gold standard if we are getting so this is the positive negative. So gold standard which is positive they are two positive. Gold standard which you are getting negative they are two negative. Okay, suppose in this is diagnosis you got certain positive here, you got certain negative. So how you can say, for example, assume that there is 1000 animals is there, among that 200 animals living and 800 that is right. Now this screening test they can identify the 200 animals with the disease because you use that gold standard test. Okay, so the radar can be shown here. So total out of 200, out of the diseases, 150 is positive, 100 is negative, and for negative 50 is diseases and it is 700 negative. So total 250 is positive and negative is 750 out of 1. So how you calculate? This is the sensitivity already mentioned here. The formula that is true positive by disease total diseases and specificity true negative by total negative diseases. Okay. So if you calculate this one, this coming sensitivity is 75%, 150 divided into 100. Okay, 150 positive divided by, notice the 100 divided into 1 is 75, and specificity will come 700 to 100 divided by 800, which is 87.5%. So this is how you are going to calculate the sensitivity and specificity of the diagnosis. So another important factor for developing a diagnostic test is your reproducibility, repeatability, and reliability. Okay, so reproducibility, that is the 3 R you can say. Okay, this reproducibility, repeatability, and reliability, they all mean that the results of a test or measure, they should be identical or they are certain. Both be similar each time whenever you are quantifying, you should get similar kind of. But there is certain variation may be there because it may depend on the level of observer. Suppose two person is working on the same diagnostic, some may observe some result, some may different result because it's to eye contact or variation. Okay, it may include changing condition like time, location. Okay, so there is certain little variation may be acceptable. What are the defined type of variation? Maybe inter-subject variation. Okay, inter means in the same. Inter-subject variation, inter-observer variation means one person can see the result 2 times, may tell that the result is same. Inter-observer variation, suppose 2 defined individual is doing the same test, so they may see different result. So inter-subject variation, suppose this is one individual and one individual. So there is between same time, same individual working in a some lab and again in the different lab working, it may be due to some, the result may vary, may be due to certain physiological, environmental condition in that. Okay. This is called inter-subject variation. What is inter-observer variation? Is a variation in the result of the test due to multiple observer examining the test. Suppose individual A and B is looking for the same, so they may say different result. And here inter means same individual A. What is intra-observer variation then? Means A is looking, okay, means same individual, they are due to some of the examining the different time. So after suppose today meeting, after 3 months he is submitting, so results may vary slightly. So this is called intra-observer variation. So these are the different common variations in occur in the result, right? So this all about the general about the diagnosis. Now coming to now our molecule. What are the defined molecular diagnosis? You know that let me say this molecular biology, once it is open, it has then the new avenue for development of different types of things. It may be developing diagnostic, maybe vaccine, maybe therapeutic, nanoparticle based therapeutic, and all this so many. So if we can compare that, what are the molecular basis? One may be looked at serological diagnosis. Serology means based on antibody. You know that antigen antibody interaction can develop the diagnosis. For example, you know that it's called ELISA, exactly, immunosorbent, okay, which are different type of pandemic like direct, indirect, sandwich. Based on that, you want to detect the antigen or antibody. Okay, so you can go for serological diagnosis. Okay. Suppose you are going to feel collecting the blood and taking the serum, then the serum go for ELISA and see that antigen is present. This is a very common. Another diagnostic is the nucleic acid based diagnostic. This is based on your nucleic acid, either DNA or RNA. Okay. Previously, they were doing by hybridization method. Now, once the polymerase chain is coming, now PCR is the most common method. And you know that is also quantified, it's available like rare than PCR and all those things. Besides this, mobile high throughput test is also available like microarray, peptide nucleic acid, biosensor, okay, point of care diagnostic, next generation sequencing. So these are the all common methods for diagnostic of disease. Now I will be coming to that whatever the works we did here in a nutshell. So just I will be explaining here and I will be showing my outcome also. We have 2-3 projects it is running our team, which has been funded by DBT, DST, than your ICMR and your National Livestock Mission and all. So our teams, we are working on different angles of development of diagnostic and vaccine. We have one project is ongoing, it is in Swinostics. It is a platform for development of validation of on-field diagnostic of important pig pathogens in India for commercial exploration. We have developed various diagnostic here. We have developed a multiplex assay, implication-based diagnostic assay. We have developed, you know that single detecting virus, it is difficult. So if you go for multiplex, in one diagnostic, you can identify multiple diagnostic. So this is that we have developed a nucleophage viral DNA isolation kit. This is the within 5 minutes today. Yesterday we got this certificate. I have forgotten to mention. So this is within 5 minutes you can isolate the viral DNA. Okay, so we have developed two multiplex assays. One we have given PGplex, P and PGplex are three RNA viruses can detect. PGplex three DNA virus can detect. This technology has been certified by ICR. Okay, so we have also developed a diagnostic assay for ER, porcine lymph product syndrome virus assay. This kit is also available with us. So some of the diagnostic assay we have developed and commercialized. Now recently we have commercialized the diagnostic. What I told you, PGplex is a multiplex. You can simultaneously detect three viruses like African swine fever, porcine syndrome, porcine parvo, and PGplex are here with the classical so Japanese ERRL. These are the kits we developed, and it has been commercialized recently by this biotech. They have taken our technology and it will be available in the market within 1 or 2 weeks. So this is the first we have developed the world's first multiplex panel for diseases. In the same test you can identify all the viral pathogens. So this is all about the nucleic acid. After that, this is a PCR based, you know. What is the difficulty in the PCR? You know that once the PCR has been completed, okay, so you have to go for agarose gel electrophoresis. You need to some instrument like your PCR machine, thermocycler. You need to see water bar. You need to see agarose gel. So these are costly equipment. And PCR is taking time. You need 3 to 4 hours to complete full course. So we need to think for some other diagnostic which are rapid, user friendly, and they are not so costly instrument you don't require. So scientists has developed a different test also, like 2 tests which have standardized in our laboratory called as LAMP and PSA. They are called isothermal assays. LAMP code for low mediated isothermal amplification and PSA code for polymerase spiral reaction. So these are the 2 different assays based on isothermal. Isothermal means you know that in PCR what you need? We need 3 temperatures, right? Denaturation, annealing, and your extension. 94, then after 50 to 60 for annealing, then 70 to 80. So different temperature. But for here, only a single temperature you can keep that because you are using certain enzyme, or you can say that like that polymerase you know that we are using for your PCR. Here we are using another kind of polymerase, BST polymerase. So they are it's very highly sensitive and they have that capacity to stand displacement. So you don't require multiple temperature also. So in that case, you don't require to go for PCR machine. You simply require to a water bath, and you can put certain dye, intercalating dye like cyberkin. You know that cyberkin can bind to your double-stranded DNA. Okay. So once the color changes will be there. So this is once it is become yellow, it is the positive. Okay. So we have developed this LAMP assay and PSR assay for detection of your African swine fever virus. Okay, and this technology also will be commercialized very soon. And it is by naked eyes you can see based on the color. You don't need to run on the agar gel and you see that. Okay, so this is the visual assay. So this is also we have further compare with the other tests like real-time and we have seen up to 50 nanogram or picogram, we can 5 picogram, femtogram level of African swine fever virus immunity can be detected by this. It has been already published in Journal of Bioscience. Okay. So we have also filed a patent and this patent not yet granted, but it has been filed. Okay. So besides this, we also develop quantitative PCR. You know that why the quantitative PCR is required? Because PCR, what is that? It is a qualitative assessment, positive or negative. But how much copy number is there? How much DNA is there? You cannot know by the PCR. Yes, you can know by PCR. You can the product you can clone it, and after that you can go for serial dilation. But this will not give you exact signal. But quantitative PCR will ultimately you know what is the exact level of the genomic DNA is present in the or the RNA copy is present in the particular file. Okay, so this is called quantitative. Like real-time PCR, you can go by to either CyberGreen or TechMan code. Okay, so based on that, you can develop the assay. So we have developed 3 diagnostic based on qPCR using TechMan code. One is for African swine fever, one is for your classical swine fever, and one is for your and this technology will also be commercialized in due course of time. These are working. So, and this technology is also available in AgriNovet, and whoever is interested, or they can show their expression interest, and the technology will be commercialized. Okay. So, we also, besides this, we also have work on certain serological assay like ELISA-based diagnostic. I have already mentioned it is either antigen or antibody interaction. We have developed a receptor. You know that host receptor is very important because this is the host receptor where the viral protein is interacting and virus is entering the code, right? So what we did, we targeted the host receptor. That is a receptor called CD163. You know that this is for HIV patient. This CD163 receptor is also important. So this HIV virus will enter to the host cell by procedure. Similarly, in P, that PRRS, that is porcine rare syndrome virus, they also utilize CD163 receptor to enter into the code. So we have targeted this CD163 receptor for developing an enzyme-linked assay for developing diagnostic against PRRS. So what we did, that we have taken from NCBI the CD163 protein sequence, okay, and of that post mean of the sequence of what we have taken, pig we have taken, cattle we have taken, then you dog, poultry, fish and human CD163 protein we have treated. Then we have taken the PRRS, that is porcine liver virus of the pig. That 2 genes, that is GP2, glycoprotein 2 and glycoprotein 4 are the 2 major protein which of the virus interact with CD163. Okay. So after that we have gone for homology modeling by using Discovery Studio. Okay. So then we have predicted that 3D structure model, gone for docking, and we have identified the interaction site of this virus protein with the CD163, which part in that host protein is interacting. Right. Then after that, what we have done, that we have identified the which. Then after that we have gone for experimental validation of that rock site in different prints we have seen using PCR sequencing. After that what happened, this whatever the rock site present CD163 we have expressed. Okay, the laboratory gone for cloning recombinant DNA technology. This CD163 has been taken from pig, we have amplified, then we have gone for LTS base, then we have using for developing the ELISA and detecting the PRRS file. How the ELISA is developed? Because first what we did, the ELISA plate, we coated with this the receptor protein which we express in the laboratory of that CD163, right? After that, we have inoculated with the antigen which you are going to detect is unknown. After that, we have given the primary known antibody against that antigen, okay, and secondary antibody which is conjugated to that your this one, yeah, and then you add the substrate. If this antibody is specific to antigen will bind, and if that antigen which is PRRS, then it will bind to that CD163. This is the your CD163. So if it is specific, then it will bind. So if this antigen is specific, antigen antibody will bind, again the secondary antibody. This is the way how this is work. So this is our certain result. We have gone for homology modeling of SARS-CoV CD163 receptor. Then we have seen that how it is interacting. You see that this is the interacting site, and we identify that exon 7 is the region of your portion CD160 is the site where the GP2 and GP4 of your PRRSB is defined. So we have taken this exon 7, and if you see, this is the interaction site we only find in case of your SAS profile. This is the interaction. This is the interacting between the two domains. Okay, but if you see cattle, boss is cattle, but it is not interacting. There is a gap. Similarly, in fish also gap. It is not interacting. So canine, dog also it is not interacting. Poultry, galas not interacting. Human also not interacting. That indicates that this PRRS virus not interacting with the other species except to what is called in the pig or suspect. Okay. So this is the sequence we analyze, and after that CD162 we have expressed, recombinant protein we have expressed, we have unified that protein. Okay. So after that this one we have used for developing the ELISA. These are the certain results we have seen. So by this around 270 post-mortem samples tested, and we have seen also not so much accurate, but only 58.88% the test was found to be accurate, and the CV value 6 to 10%. But this is then the way we can develop this diagnosis. Okay. And we have also seen the species sensitivity. You see, it was positive only for PRRS, not in the other disease like classical swine fever, CTP, Japanese influenza was negative. So this is important. Another angle we are working on the point of care diagnostic. You know that lateral flow assay. You know that the pregnancy diagnosis. What we are doing? Some kids it is coming to the household for seeing that they is pregnant or not. Like urine sample we are getting. If we are getting 2 lining is positive, if we are getting 1 lining negative, that is called lateral flow assay. That is also based on antigen activity. What is the principle in that lateral flow? Why is that the flow is in the we will move laterally, not vertically. Okay. So what happens? It is consist of your sample pack followed by conjugated pack followed by it is a nitrocellulose membrane. There is a test line, control line, and then absorbing. So what happens? Sample pack, once you add the sample, the antigen will come here. The conjugated pack, what you used to do here, the antibody against that antigen, you need to conjugate with certain nanoparticle, like gold nanoparticle or silver nanoparticle. You conjugate, okay, then you put it here. So what will happen? And in the test line, you have to go with the specific antibody against that antigen, and in that conjugate pair, same antibody only, but it is conjugated with the nanoparticle, which will give the color here. And in the control line, you give anti-antibody, which will bind against that antibody. Okay. So how this will work? Once suppose the blood sample, the antigen will come. So the antigen, it will have some specific reason. The epitope will bind to the epitope-paratope interaction with the conjugated antibody. So once it is binding, so it will flow towards the test line. And in test line, again your antibody is there, but it is unconjugated. So this antibody will also try to bind. Once it is trying to bind, it will give a color reaction. You will get a line. Okay. And for control line, after that, whatever the remaining antibody is there, it will that sample is there, it will go toward the test. Control. And control line having anti-antibody against that antibody. So unbound conjugated antibody which is there. Okay, some of the unbound will come to control line. This will bind to that anti-antibody. So this once it is reacting, it will give you the line. And remaining will come with impact, this will be absorbed. So this is the way how this lateral flow is work. So we have developed a peptide-driven lateral flow platform, means using classical swine fever. What we did, classical swine fever virus we have taken into peptide. We have not expressed it laboratory, but the peptide sequence we have taken, and you know that the peptide is a small protein, so it will not give you immunogenicity. So we have conjugated with a certain hapten called KLH. Okay, small molecule, because you know that for antigenic response you need to be a glycoprotein or larger protein. So for that we have added the KLH. Okay, after that we have immunized the animal we raise the hyperimmune serum. That serum we have tested for ELISA for it is working or not. After that, that antibody what we got, we have purified that antibody and then we have conjugated with the nanoparticle and we have taken from mango leaf, we have taken the silver nanoparticle. Anything you can take the some other leaves also, you can take the gold also as per your research. Okay, we want to see that the green nanoparticle can also be utilized for development diet. So after conjugating, similarly what I have explained here, put in the lateral flow assay, the conjugated fat protein, that then after that once you loading the sample, if you getting two lines, so it indicates positive. And if you getting one line in the control line, not in the test line, means if control and test is positive, that means your case is positive. If only control is positive, test is negative, that is negative. So this we have developed, and this is the peptide we have two peptide we have characterized. We have taken the T cell epitope. This one, you see this peak, it is yellow peak, it is indicating this is the high reactogenic index. So that part we have chosen. Okay. So after that we have seen the antibody titer after immunization in rabbit. Okay, and see that day 21 the antibody titer was higher. So followed by this is the protocol how the nanoparticle we have synthesis from the mango leaves. Okay, the silver nanoparticle, and followed by we have characterized that conjugation by UVB spectrophotometry analysis. Okay, we have seen that it has been right shift of the lambda max from 521 to 5, so indicate that has been conjugated. Followed by we have gone for zeta potential analysis to identify what is the size of that particle, nanoparticle. We have seen it is 31 to 38 nanometer. Okay, and after conjugation the size has been increased from 156 to 143 nanometer in size. Okay, so followed by this is the that assay which we developed. You see that test and control line here, two line is here, and then the control it is negative. So this is the two lines we have seen in defined serial diagnosis, the sensitivity and basis. We have seen in means up to 20 nanogram per piece of CSFP antigen can be detected by this assay. And we have further specificity also we have seen, see only the CSF virus we got that two lines, but in other viruses we did not get any test line. So indicate it is very specific for CSF, not other. This is the diagnostic, and yesterday this paper has been accepted and it has been published in the Journal of Immunological Method. It is in 4 stage. So beside this we have also used for developing for the African swine fever also. We developed a kit called PQ-Chrome ASF. This will be coming to the market. So this is for Africa and it has been filed patent also here. So this is the name we have given PQ-Chrome. And this is all about our diagnostic. Beside this, we have also one research project on FAO fund. That is from this is a foreign funded project of All India Network Project of Antimicrobial Resistance in Food Animal. These are the some of my students as well. They have published, they have done for whole genome sequencing. Okay, some of them know that antimicrobial resistance also is one of the important factors. Okay, we need to identify what are the antimicrobial resistant genes are present, what are the antibiotics. So our project is working on this. We have gone for whole genome sequencing to identify that antibiotic resistance in the fetal waste. Then we have also seen certain transposable elements which are responsible for transmitting antimicrobial resistant phenotype. We have gone for certain actually beta lactamase from. Then we have used the CRISPR-Cas method also for knocking out certain antimicrobial this one. It's called antimicrobial resistant genes in the milk cow. This paper is part. So besides this also we develop certain diagnostics for detecting antimicrobial resistant vectors. You know that some of the antimicrobial resistant like E. coli, MRSA, Staphylococcus aureus, they are most common, Klebsiella and all. So we developed a universal primer set for detecting it. The single set you can detect E. coli or Klebsiella because they are very closely spaced. It's very difficult to identify. So this we have filed patent also. We have also developed a multiplex assay for simultaneous detection of E. coli, methicillin antibiotic and methicillin sensitive. This also the commercialized soon. Besides this, we also developed a herbal aided AMR bacteria tester, that is AMR tester with a culture fee, because for detecting antimicrobial resistance also sometimes you need some harsh chemical and all. We developed a assay using certain plant extract, okay, and based on the changes in the color, okay, we can tell that what the level of the antimicrobial resistance bacteria is present. Okay, so this is also under evaluation this test. So besides this, we have also worked on detecting E. coli. So that paper is published in LWT Science and Activity. This was for detecting in the cow milk, in milk DNA, that is master milk DNA, whether E. coli is present or not. We have developed a assay. We have by using certain nanoparticles. So we have coated the nanoparticles and certain probes we use. Okay, and based on the colors or magnetic wash. So if you get a droplet here, it is indicated positive, and if you get a transparent, it is negative. So this is the way we can develop, and we have seen that up to 100 picogram of the this E. coli DNA can be detected by this assay. Okay. Then we have also used the lateral flow assay for rapid detection of the Staphylococcus aureus. I already mentioned the lateral flow. We have used for Staph aureus also. So I am not discussing it here again. So these are the certain granted patents. Recently, this is some patents also. So now coming to the another project which we are working on the vaccine. That is the development of multi-serotypic virus-like particle-based candidate against Indian isolated porcine virus, which has been given by DASD, Department of Animal Husbandry and Ag. So you know that in our country, the commercial, I have already told, porcine circovirus is most immunosuppressive disease. So there is no commercial vaccine for this porcine circovirus. You know that porcine circovirus having different strain, that is PCV2A, 2B, 2B, PCV3 also coming. Okay, in India the most prevalence is your PCV2B, and although means previously PCV2A and 2B vaccine has been developed. But it is shown that these two vaccine is not protective against the PCV2D which is present in animals. So these are the some photographs of our farm. In fact, in the porcine circovirus, you see that some of these animals have died. This is the embryo. Okay. So far we don't have any indigenous vaccine, and for exporting the vaccine, it is a very cost effective. Based on that, we have taken this project, and what we did, we have synthesized the porcine circovirus to the Indian isolate strain. We have grown into a open vector, and we have expressed in the baculovirus system. You know that by certain insect cell we can express it. So this is the expression how we did, and this will produce a particle called virus-like particle. It is not virus, because what is the difference between virus-like particle and virus? Virus-like particle, they don't have nucleus, only the capsid particle. So it will not infect, not multiply. Okay, only the capsid protein will be just like a virus-like structure. Okay, okay. So this is the, you see that how it is in microscope transmission electron. We have used the S-alpha odoratum AU38 cell line, which has been exported from the UK for developing this virus-like particle, which has been confirmed